crops as well. It also has the ability to disperse over long distances, so it's capable of flying over 70 miles per day. And as well, it can be an urban nuisance overwintering on human structures. I actually had, I think, 3 of them in my house this winter, which my cats found very entertaining, but my husband found less so. So how you can identify BMSB, we do have some native stink bugs that look similar. Some key characteristics you can look for are the shield shape. I get a lot of questions about squash bugs and if they're the same or not. So one of the differences you can look for is they have much more of a shield shape. Look for those 5 points versus squash bugs are kind of more oblong and oval. You can also look for these white bands on their antennae and then kind of that striping pattern around the outside of its body. BMSB overwinters as an adult, and so in the spring, they will start to emerge and lay some eggs. They will have nymphs that look similar to their adults, but can't fly, and then they'll eventually develop into adults. And usually they'll lay some eggs there in the summer. We'll see some aggregations in the fall. That's when they'll start getting into your houses, and then they'll overwinter and start the whole cycle over again. So BMSB arrived to the US in the mid-1990s on the East Coast, and then we've quickly seen spread. It's now found in 47 states and 4 Canadian provinces. We first saw it here in Utah. It was detected in Salt Lake City in 2012. We then had some establishments and some nuisance reports in 2016, and then the first reported crop damage happened in 2017. It's now established in quite a few counties and detected in 2 more. And as I mentioned, BMSB is such a severe pest because it can feed on so many crops. I'm just going to highlight a couple of those that are significant for Utah. So we have peaches. These are a pretty important crop for BMSB. It can complete its full life cycle on peach. And then we also have apples. It uses its straw-like feeding tube or a proboscis to suck nutrients out of that fruit. It can cause that cat-facing damage that you see on the peaches, and then corking on that discoloration, kind of bruising on the inside for those apples. And then lastly, I want to point out tart cherry. This is a pretty important crop for Utah, and my colleague Zach Shum conducted research that found that tart cherries are highly susceptible to BMSB damage, and early feeding will actually just cause those fruits to drop. You won't see quite as much damage. You'll just lose a lot more crop out of it. So a lot of research has focused on trying to manage BMSB because it is such a severe pest. And so to do that, we have to take some different tactics of timely monitoring and trapping. Those help us know when and where populations are present. A very common control method is broad spectrum insecticides. However, these have pretty low effectiveness. I talked about the ability of the stink bug to fly. And so oftentimes the stink bugs will fly away and eat something else and then just come back with those insecticide sprays not having a whole lot of effect. These can also be very costly to growers and disrupt IPM projects. So causing some other issues in terms of other pest breakouts as well. They're also not great in terms of they can kill off quite a few beneficials. So recently, a lot of research has focused on the idea of biological control and using predators and parasitoids against BMSB. So I'll show you there are quite a few predators and parasitoids that can attack it. The most vulnerable life stage is this egg life stage. In Asia, populations are kept very well in check by these tiny, tiny parasitoid wasps. And I told my mom for years that I researched wasps, and then not until I showed her one in person did she understand that these are very small, about the width of a nickel is what I tell people. She said, those aren't wasps, those are gnats. But they are little but very effective, so they will actually sting the stink bug eggs and lay their own egg inside, killing the stink bug as the wasp develops, and then it will emerge as a full-grown adult. So in Utah, we actually do have some native species that can attack BMSB. I'll show a couple of them here, but unfortunately, these have not traditionally had great rates of success. They are not evolved and adept at attacking those stink bugs. So rates of emergence coming out of those eggs is typically pretty low, of about only 0.5% to 3.7%. These wasps do serve good purposes where they control native stink bug populations, and they have much greater success on those. But we've also been looking at Trisulcus japonicus, which it has a very cool common name, the samurai wasp. But a lot of our native Trisulcus don't actually have a common name, so I typically stick with the scientific name just to keep an even playing field. But I will call it TJ just because it's easier to say. This wasp underwent testing in quarantine facilities to try out its potential as a classical biological control agent, and see if we could do some releasings to help control the pest. But then it actually arrived to the US on its own, likely through a parasitized egg mass being transported. That hasn't been confirmed, but that's our best guess. So that was in 2014. We detected the wasp here in 2019, again in Salt Lake City, Utah. And some early research on the wasp gave good support. We noticed that it was parasitizing just over 20% of BMSB and had much higher emergence rates than any of those wasps, native wasps that we have. However, we do have some environmental challenges. The Intermountain West poses some unique challenges. Compared to Asia, we do have high elevation, which also gives us some heavy snowfall that the wasps aren't necessarily used to. We also have arid hot summers, and some recent geographic modeling determined that our area specifically is only marginal or unsuitable for establishment. And so that kind of leads to the research that I conducted as a master's student and what I'm going to talk to you about today. So to really understand the status of TJ and work towards its conservation here in the Intermountain West, I had 3 objectives that I worked on. So the first was to look at landscape effects on native and exotic Trisulcus species. The second was to look into the possibility of using stink bug chemical lures to attract parasitoids. And then the last is I just wanted to learn more about the movement of TJ in the Intermountain West by surveying for it in some new places. So I'll start off with objective 1. And again, this was just to understand some different factors affecting the abundance of both our native wasps and this exotic TJ or samurai wasp. Specifically, I was looking at the factors of urban surroundings, orchard ground cover, and then the stink bug presence. So we did this by doing yellow sticky card surveys. We deployed sticky cards for 2-week intervals for a couple of years. And I'll thank my colleague Cody Holthouse for also letting me use some of his data to help with this. So from the photos I showed you earlier of the wasp on the dime, you may have already gotten a feel for how small these wasps are, but again, they are so tiny. Those are what I have circled in those blue circles. Very small, and so they're pretty hard to detect and to understand their movement, but sticky cards have been found to be very attractive, and so that's what we used. We surveyed 24 to 36 sites per year, just depending on the year. And then I used some very cool data on Cropscape to look at how urban land cover was affecting some things. So I looked at what the makeup of the area around was in a 1 kilometer buffer. So I quantified this into some categories of low, medium, and high. And I want to point out here in southern Utah County, this patch of hot pink represents kind of an area of important crop production, and we have quite a few orchards there that we wanted to look into. So what we found, now we get to the good stuff, we get to talk about what we learned. So what we found is that in 2019, we mostly saw this wasp in close proximity to where we originally detected both it and the stink bug, which kind of makes sense where it's following its host. In 2021, we saw some expansion southward and some increasing numbers. And we were just noticing, trying to figure out what the movement of this wasp was, if it was able to spread or not. You'll notice that we still don't have super high numbers, but in 2021, we're seeing, again, increasing numbers. And we did shift this year. I wanted to look more into those areas where we have those orchards of concern. So we did a lot more surveying down south, and we switched up some of our survey sites to make sure we get an understanding of where this wasp is in terms of where it can be most effective for control. So looking at how urban surroundings affected our TJ, we saw a pretty clear trend across the year. And we had increased biweekly captures each year, which was good to see that the numbers are increasing. And then we saw that pretty clear trend where we're seeing more wasps in those highly urban areas. We had a couple different theories about why that might be. So it's likely due to the presence of the host stink bug. There's been a lot of research that's come out recently talking about the importance of those structural buildings for the stink bug for overwintering. So it's likely that it's following that host insect. It also could be because of the urban climate is a little more sheltered, a little more protective and mild. And then one of the last thoughts is that these wasps, while they do attack the stink bug, that's not what they're feeding on. They still are reliant on floral resources, so they could be taking advantage of the floral resources and more of the diversity that these urban areas provide. We also looked at this for our natives altogether, all of our Trisulcus, and we didn't see quite the same trend at all. It was a little bit all over the place. We didn't see that clear trend. So maybe suggesting that our Trisulcus aren't reliant on BMSB as a host, or maybe aren't as reliant on those urban resources. So that leads us to talk about a little bit more about those floral resources. So urban areas often have ornamentals. They have a lot more diversity. Agricultural sites, on the other hand, are often very limited for parasitoids and pollinators. There's not a whole lot for them to eat a lot of the times, not a whole lot of shelter either. And so one of the things that we can do to really support that is to increase our strip cropping or border cropping. And I wanted to look at some of the effects of that. Some of the conservation applications that we have are to provide nutrition, habitat. These, of course, can extend the lifespan, and also they've seen some studies where there's actually increased offspring production as well, depending on what floral resources are nearby. And again, these wasps are very tiny, so it's hard to really know what they're eating or what they need. And so that's kind of why I wanted to look into this topic more, get a better feel for how these wasps interact in their environment. So to do this, I deployed these yellow sticky cards at 3 different types of orchards. We had 8 sites of each. So we had bare orchards where the ground was just dirt, cultivated, no floral resources, and then we had well-maintained turf grass orchards. Lori will laugh because this is her orchard, and she says that it's not always well-maintained, but I did my research, it looked pretty good. And then we also had floral orchards, and these included unmanaged sites that might have had weeds or wildflowers. This also includes sites that had purposely strip cropped or border cropped flowers as well. Pretty much anything that had flowers that might sustain the wasps. So this is kind of a crazy figure I'm showing you here because we learned a whole lot from this study. We analyzed the abundance of each group by the number of captures per that yellow sticky card, and we looked at this across the season measured in degree days. And so degree days are a common way to measure the time and temperature, and this lets us standardize it across many places and many different years. So it gives us lots of seasonality information. So the first thing I want to point out here is that we had very low numbers of both BMSB and TJ. So we had up to 3 per card on average. But then I'll also point out here that the TJ, or our exotic wasp, is tracking BMSB very well. It's following kind of those peaks and dips that we see, which is a good sign to us that it's doing that. It's very reliant on its host and is probably being effective there. So when we analyze the differences between these ground cover types for Trisulcus japonicus, we found that there were significantly more TJ in our bare sites than floral and turf. This kind of threw us off a little bit. We were expecting floral to be, you know, the big winner by a landslide here because it has those resources. But there is some caveats here. If you notice, I have quite a few more stink bugs in our bare sites than I do in our floral and turf grass. So there could be a pretty heavy bias there in our results where these wasps are following their host regardless of the ground cover. But we need some more research to parse that out a little bit more. We also recorded the captures of our Trisulcus in general to see what was going on. And I'm glad we did because it gave us a much bigger sample size. We had up to 60 individuals per card. So that lets us do some better statistics. So when looking at that, we analyzed the season-long abundance and we didn't find any significant differences. And that didn't quite sit right with me because you can see we have a pretty sharp peak there at the end. So I took the analysis a little bit further and looked at what I'm calling peak season. So that's after 900 degree days. So kind of looking at when they're most abundant. And then we did see that heavy significant come out of the wasps have a heavy preference for floral sites, which that holds true to kind of our hypothesis and what we were predicting. So looks like they're taking advantage of those resources. And then last I want to point out here where we have BMSB on top and then our group of all wasps on the bottom with our natives. And we don't quite see that beautiful alignment with the peaks and dips. It looks like our native wasps might be more abundant late season and might not specifically line up. So that gives us a lot of ideas of who might be the best biological control option. So when all said and done, looking at this, our floral sites had about 54% of all of our wasp detections. So pretty strong evidence there that floral's doing something right compared to bare and turf grass. And then we also wanted to split this out by species and look a little bit more at what's happening. You'll see this also gives you a good comparison of the numbers of wasps that we have. So T. japonicus, our samurai wasp, there is that dark purple. And so it made up only about 4% of what we were finding, so not a huge portion. And then our one native that's called Trisulcus euschisti is how you say that. It's a real fun word. Made up about 78%. So a pretty large portion of what we had. But we did notice that most of our natives are following a similar trend where they are more abundant in floral. Only our Trisulcus japonicus was kind of throwing things off where was more present in bare. And again, that could be due to the bias, or it might be utilizing those urban resources and ornamentals more than necessarily our Utah wildflowers. So what does this mean for Utah? Overall, we had very low abundance. Again, it was only detected in 2019, so there could be some implications there in terms of if we're going to see higher numbers or not. But again, it's not the ideal habitat, so we'll have to see. But we did get some strong evidence for TJ utilizing urban resources. Again, it may be going outside of just that orchard and utilizing those urban resources in that 1 kilometer buffer. We did learn a lot about this wasp is tracking its host very well. which is good news in terms of it's probably not going after our other stink bugs, so maybe less target effects. So we learned quite a bit about that. We also learned that floral ground covers are very beneficial to our Utah natives. And so even regardless of what's happening with the samurai wasp, having those floral ground covers can help support our native wasps, which do still provide a level of control. And then lastly, I talked about degree days with the last graph, and that really helps us understand the seasonality of these parasitoids, where they all are small and hard to know what's going on. It's good to get a better understanding of when they are most active. That can give us helpful information in terms of when we want to possibly release these wasps and have the best success, and also where we might want to do that. It also gives us information about timing our pesticides and insecticides well, to make sure we have the least damage and effect to them, and also when we can plant and provide floral resources to them. So I know that's a lot of information, but we're now on to objective 2. So I'm still going to throw more at you. This took 2 years, so there is a lot shoved in here. So my second objective here is to look into the idea of using stink bug chemical cues, which are, in this case, we're calling chiromones, to attract parasitoids. That big word chiromone just means that it's benefiting, those chemicals are benefiting the receiver, which in this case would be the wasp. So all those chemical cues that the stink bug or their eggs are putting off, attracting these wasps. Kind of a fun idea. Some other people have looked into these different chemicals and determined some specific chemicals. We won't dig into their scientific names and go too far in. But we looked into taking these chemicals and putting them into some novel lures. We used rubber septa lures, much like many of you might have used for possibly doing pest management, where there's pheromone lures, a very similar idea there. And so we made some different ratios based off of other research to try and determine what might be the best to attract these wasps. So it's kind of a weird idea, but there was found to be one chemical that's specifically an attractant, and then another that was a repellent. But some research has found that if you actually put these together, you get better results than using the attractant alone. So that's kind of the reasoning why we tried some of these different things. So we, of course, had a control as well to help us understand what was going on here. So I took these out into the field. We had a site in Salt Lake that we were using. We used catalpa trees. These are a big host for brown marmorated stink bug. And so we find lots of stink bugs and lots of wasps there. So we thought that would be a good plan. So essentially what we did is we took each of these lures, attached it to a stink bug egg mass, and then we put that out in the field for a few days, and then came back, picked it up, and we got to see what happened. So we were looking to see if any wasps had parasitized the eggs, and if they had, what species those wasps were, and basically how successful that they were. And here is what we found. Again, lots of information here, but I'll run you through it. So we're looking at these different treatments of the lure. So each of the bars represents a different lure that we tried. And then we have the control there as on the left as the kind of the baseline for what's going on. So we saw that our 100% attractant lure was actually the best in terms of having parasitism. And then our 90 and 80% were not as great. They did not as well as the control. So this kind of threw me slightly for a loop. Maybe that other research isn't holding true in our situation, where maybe the repellent is repelling. We weren't quite sure, but what you can see here is that we had the best of Trisulcus japonicus or samurai wasp emergence on that 100% treatment. You can't really see the Trisulcus urschistii emergence because it's one tiny little bar on that control. And then we were also looking at all these different things that can happen to the eggs, just to understand if we're possibly attracting other predators, if we are somehow kind of damaging those eggs in some way, what's going on there. So we kept some baselines. We measured how many stink bugs are coming out as well. So lots of fun stuff there. So what we found overall here is I put out 92 egg masses over the course of the summer that I did this, and sadly only about 13 of those ever showed parasitism, which is pretty low rates, which makes it, again, hard to do statistics when you got low rates like this. But what we saw is that Trisulcus euschistii and Trisulcus japonicus, our exotic, went after about the same amount of egg masses. So one tried to parasitize 7, one tried to parasitize 5. So fairly evenly matched, but then we also saw those emergence rates, and Trisulcus japonicus came out of a much larger portion of those. So it emerged for 5 out of 5 instead of 1 out of 7. And then looking at those by that egg unit itself, we saw that Trisulcus japonicus had an emergence rate of about 78%, so doing pretty good. Close to what we see in Asia with it, not quite, but very similar. And then Trisulcus euschistii, we had a sad little 1.6%. So even though it attacked 7 egg masses, we actually only had 3 wasps come out of all of those eggs that it attacked. So just a little bit of what's going on. We got a lot of a better understanding of what's happening biologically out in the field. We did have a very interesting situation where one egg mass had evidence of parasitism by both wasps. These wasps will guard their eggs, and we had one species guarding and another species come out. So that has quite a few interesting implications in terms of their interactions and competition. So that's research for someone else to test, but we'll see. But some really interesting stuff going on there. I like these pictures because it's a good example that I get to show you of what we might see. The picture on the left is a Trisulcus euschistii. She's guarding those eggs, but they are still bright green, and they have those little black triangles. Those are called egg bursters, and those only happen when stink bugs are about to come out. So they're not on all of them, but on quite a few of them. So it's likely that we might see 1 to 5 wasps come out of this egg mass, if any. And then this other photo is a good photo of what parasitized eggs look like. It looks like the female did a pretty good job on this one, where all of them are turned dark, and it's likely that we're going to see quite a bit of wasp emergence out of those eggs soon. So I talked about only 13 egg masses out of 92. It's hard to do stats, hard to get some good evidence of what's going on. So our next steps is we actually took this back from the field, and we wanted to look at the stuff in the lab where we could supply wasps. This lets me put as many wasps as I want and see what their interactions are. So we did this using my very fancy box that my colleague built me. I was very grateful for it. But essentially what we have are these clear sticky panels on either side. There is a lure attached to either side. We tested a control versus all the other ones. So I put in, you'll see there's a little cork there on the bottom. I took that cork out and we inserted some wasps, usually 4 to 5 of them. And then we let them do what they wanted for 30 minutes, and we got to see what was happening. So we considered it a choice if they stuck to one of those sticky cards on either side. So again, we wanted to test these lures that I tested out in the field. We tested each of them versus a control on the other side, just to see if we had some attraction differences. We also wanted to look into the possibility of my lures being too strong. There's a fun thing where if you actually load the lures too strong, they can end up being repellent in general. And where I had very low parasitism, I thought maybe that's the problem. Maybe we'll take it down a notch. So we actually also tested a 5 milligram load rate lure. And we just did this with one type where we just wanted to test that 100% attractant to kind of get an idea of what was going on. So to explain this graph, what we would expect to see if there's no differences is what you see on that very first set of bars. So if there's no differences, if the wasps don't seem to care between the control and the treatment, you would see an equal choice, which happened with our 5 milligram lure. So that's our low load rate there. So we didn't see any significant differences there. That next bar you'll see is our 10 milligram 100% treatment, and we did have significant effects where wasps were more often going over to that side and choosing that lure. We also saw this weird thing where the 100% was attractive and the 80% was attractive, and that 90% was not. It was a little bit, but not to significance with statistics. So we maybe need to run some more trials to understand what's going on there. It's kind of a curveball, not sure quite what's going on. It could be that these are just the best ratios to use because they best match what a stink bug is actually putting off. But definitely some more research to look into there. So what this means is that we now know for sure that we have Trisulcus japonicus parasitizing much more than our natives, and they are much better. We had some little bit of preliminary research on that already, but we've got better evidence now too. We understand that Trisulcus japonicus is kind of a much better bet for us in terms of what we should use as biological control. And we also saw only these two species were parasitizing. We have other species in Utah that you saw way back on that other graph, but none of them were attacking the eggs in this case. We learned that our high load rate is a good option, at least in the environment we tested it. Next steps might be to take that back out to the field. There's always back and forth. We got to try every different environment. We could possibly even test a higher one. It's possible that they just need that much more in the field where it can disperse farther. But we learned some good options, and mostly this was a very good proof of concept to understand this is a viable approach. Some of the other approaches with these chemicals out in the field haven't gone well, so this might be some nice groundwork first steps for that project for someone else, not me. So if we can get these lures kind of in a better state, we can get some increased targeted parasitism, possibly putting these lures in orchards or home gardens where we see a lot of the stink bugs, try to pull them to the places that we need that control. It also can help us with this problem if it's hard to know what's going on with the wasps. If we have something that can attract them, we can possibly better understand their detections, where they actually are and how many of them, and kind of true parasitism rates. Typically putting out these egg masses from the lab, we don't see great parasitism rates because we've touched them. They're possibly missing some of those chemical cues. So these lures might help counteract that idea. If you have more questions or want to read a very scientific, full of, my mom told me it was hard to read, so sorry. But if you want to learn more about this, I actually just had all this research published, my first published paper, so I'm very excited about it. But it was published in the journal Insects, and it is out there for you guys if you want to learn some more about it. And then I'll talk about my last objective here quick. This one was a fairly simple one. We just wanted to see where else the samurai wasp might be. So specifically looking around this Intermountain West area, Oregon, as well as California, Utah, and Washington have all reported these agricultural and nuisance problems. Idaho, however, has detected BMSB, but hasn't really upgraded that status further, though from my parents live in Boise, Idaho, and they've seen quite a few stink bugs on their house. So I was curious to see what's going on. And these other states around have also reported the samurai wasp. So detections are showing up. That's what those little dots on the map are. So we've seen quite a few detections, but I wanted to kind of look at where Idaho might fit into that picture, where it's between some of these states. And some other reasons that we wanted to look at Idaho is a recent paper came out talking about climate change and what that might mean for this stink bug. And it found that within coming years, there's going to be they're predicting a 70% increase in suitable habitat. That means there are a lot more places that we might find a lot more stink bugs. They specifically in this paper mentioned the Treasure Valley in Idaho. And I wanted to look into possibly this idea where the stink bugs are moving inland farther further from the coast, kind of expanding what they're doing. So where we were in Utah, I thought that Idaho was a good bridge. So we did some surveys here using those yellow sticky cards. We just threw them out places that we had growers. I'll mention some of the growers that we've worked with in Utah also have orchards in Idaho, so they were nice enough to let me put some stuff up there. They're also very interested to see what's going on. So I did this in the year of 2021, and we actually did detect the samurai wasp. This was the first detection, so a state record for Idaho here, and you'll see the red triangles are places that we did detect them. And then I also have a little pie graph here just showing what some of the other species that we have are as well. Nothing too different from Utah, so it was interesting to see. There's a little bit of climate differences, so seeing what we have in different places. We also, while we were deploying these sticky cards, we actually did find some egg masses, and we collected those, and then later we had some samurai wasps emerge out of them. So we had some direct evidence of the samurai wasp parasitizing BMSB in Idaho. So kind of interesting stuff. We collected 5 egg masses, and we saw some pretty fairly high rates of emergence of 62% from Trisulcus japonicus, but we also did not see any of our native wasps emerging. So that was some interesting information that we got there, but we know that samurai wasp is active in Idaho, so kind of an exciting discovery. That got to put us on the map there with that yellow square. Some implications is that we are seeing some potential for biological control. That might be very helpful in the coming days if some of those predictions hold true where the Treasure Valley might be vulnerable. This also shows some support for release and redistribution with Idaho, and with some regulatory processes. Knowing that it's already there makes a big difference in some of that red tape. We also learned about the lack of control by our natives going on. And then we also sent these specimens off to an analysis that looked at if these populations are different from Idaho and Oregon, and there was some interesting information there. A lot more research is going into that, but some of that analysis might be suggesting that TJ is in fact moving in from that West Coast and might continue to do so. Kind of some interesting stuff. Also some evidence that there might have been multiple introductions, that the population from Idaho might be different from the population in Utah. So some interesting stuff here, but not my, I don't have time to dig into it. So someone else will have to tackle that one. But overall, some of the things that we found is that with all my different objectives, all my research, some of the highlights are that our exotic samurai wasp is demonstrating a strong native host association with BMSB. So in terms of invasive species, they might be a very good control option for BMSB. And then we learned about the benefits and the importance of floral ground cover. So that's something that I can tell to the master gardeners and other people that I teach, that some of these things can be very helpful to increasing that diversity and increasing wasp resources. With objective 2, we learned about what's happening out in the field a little bit. Then we also learned what happens in the lab. So we got to learn about how these lures work, and we got to prove that kind of proof of concept for these lures. And now that my research is published and out there, hopefully other people can build on it and really get a functioning lure. Would be a very cool thing to have. So we'll see what happens there. I'll keep following research and see. And then our last objective is just that discovery of samurai wasp in Idaho, and some interesting stuff in terms of what it's doing actively and where it might be expanding to. So some of the things that I get asked after telling you all of this is, what can I do personally? And some of those things are to use fewer insecticides in general. Use them where you need to, when you need to, but try to keep it to a minimum. Some other research, not mine, but some studies have found that specifically spinosad and sulfoxaphor can be particularly harmful to samurai wasp and native Trisulcus species. And then we'll see with some future research if increasing floral diversity also benefits samurai wasp. Some other studies have found that specifically buckwheat, cilantro, and dill can be very beneficial in supporting these populations, giving them the nutrients that they need. So you can do that with some leaving wildflowers. You can also plant some flowers. The strip or border cropping are great options as well. And then lastly, what you can do is if you see our wasps, these little tiny guys, especially on an egg mass, leave it alone. Let it do its job. If you remove it, it will not have that opportunity to reproduce and spread. If you leave these egg masses that are parasitized alone, you'll have likely 28 more wasps to come do a good job. So that's kind of the best thing that you can do. And then lastly, I'd just like to acknowledge I had lots and lots of funding. People across the United States have been working on projects for this invasive BMSB. So I got to collaborate with a lot of cool people, get a lot of grants and work with a lot of good connections came out of this. So, and with that, I'll let you watch my fun video and we can do some questions.
Hey, thank you, Kate. That was great. So there are a few questions and some of them I answered. Okay. And so we'll focus on the ones that first I did not answer. And then if we have time, we'll go back to the ones that I answered to see if you have anything else to add. So one question, any idea of why Trisulcus euschisti has such low emergency success?
Yeah.
Success?
Probably the best reason I can tell you is that it hasn't co-evolved along with BMSB. It does very well on its own stink bugs that it's kind of naturally evolved alongside. So that's probably a big part of what's going on there, is it just doesn't quite have the ability to get past its natural defenses. So I'd say that that's probably the biggest thing there, is it's not made for it, but it still does its best. We'll take it.
Okay, the next question is, are there any potential concerns or side effects with increasing the populations of the samurai wasp, particularly as it is an invasive species itself?
Yeah, that is an excellent question. Like I think I mentioned that this wasp was in quarantine facilities for quite a while. They were looking at the possibility of doing releases before it showed up on its own, but a lot of testing has gone into that. They've looked at a lot of non-target effects. Is it going to attack other stink bugs? Is it going to attack other insects? Is it going to outcompete natives? So there's been a lot of research. The general consensus is that it's not too much of a concern. It is pretty host specific. That's what a lot of my research is helping to confirm, that here in Utah specifically, which is nice, is that we see that strong host association where it's not going after other things. We haven't reported anything here in Utah where we've seen it go after any natives, but on the other hand, most native stink bugs are also considered pests because they can eat plants. So if it does, there's not as much concern, but that's one of the big things with biological control, is to really get a lot of that research done. So I think that we have a good basis that we're not concerned at this point. I think there's a lot more to be explored in terms of the competition on if it might be harming possibly the native wasps in some way, but that's something that I had an undergrad working on a project to look into that a little bit more. And I'm sure some more people will continue to tackle that.
Okay, the next question, what are the defining characters of TJ versus the other native species?
Sorry, I could tell you that, but it wouldn't be very helpful to you unless you had a very high power microscope. Learning to identify these wasps is one of the hardest things that I've done. one of the defining characteristics is if they have 2 little mustache hairs or 4 little mustache hairs, and you have to get so up close to them. So I don't know with the naked eye that you would ever be able to tell the difference between any of them, but.
Kind of one of my questions, a follow-up, is, so if looking at an egg mass that's been parasitized, can you determine if it's been parasitized by TJ or one of the natives?
I can't for sure determine. I can take a very educated guess based on if those eggs have turned dark, based on the proportion that have turned dark. If like 70% or greater have turned dark, I could tell you with a pretty sure certainty that it's probably the samurai wasp, Trisulcus japonicus, coming out. If you have like 3, 4 that are dark, it's probably one of our natives. We have had some of our other natives show emergence, but again, it's pretty low. So I would just base it off of how many are dark is what you're gonna see.
Okay. The next question, can you talk more about the genetic analysis of Trisulcus japonicus? Do you know if it was traditional DNA barcoding or a nuclear genetic method?
Ooh, okay. So I sent this stuff off, and this is not my area of expertise, but I'll see if I can tell you it correctly. They looked at the microsatellite markers. Pretty sure that's what they're called. They also have done some genotypes in the past with other stuff with across the US. They've been looking into these different populations, but they want some more samples to look into it more. But our samples from Idaho were clustering with some that were in Oregon, but not with those that were in Utah. So that might suggest some different introductions of some different populations, but there's some really interesting stuff there that I would love to dig into further in terms of what that means with their cold tolerance and, I don't know, the different strains that we might have.
Okay, I'd be interested to understand early controls for adults before they start laying and the timing, whether that is fall treatment or early spring treatment. Also, can we use lures to attract TJ, Trisulcus japonicus, into our yards, and where would we get it?
You can't get them yet. This is pretty preliminary research on my part. I worked with the company Trace helped me. They were actually very nice to help me develop these lures and donate them to my research. But someday maybe that could be a possibility. You'll have to email Trace, keep bugging them, and maybe they'll see that there's a need and want for them. But maybe eventually that will be a possibility to use those lures. Can you read me the first part of the question again?
Yeah, so I'd be interested to understand early controls for adults before they start laying and the timing, whether that is fall treatment or early spring treatment.
Yeah, that's a good question. If we're talking about in terms of your house, some of the things I can tell you, your best bet is to seal your house up. That's the best bet for a lot of nuisance pests that are coming indoors. And then for early spring, in terms of treatments, Lori's probably going to be a better bet in terms of telling you what insecticides are best to use when. We also, I know we have the lovely Traps website online that can give you some good information about timing, but